Number 1. Symbion Pandora, the lobster lip parasite. Every year, commercial trawlers across the North Sea, the Kattegat Strait, and the Atlantic Shelf haul in tens of thousands of tons of Norway lobster, Nephrops norvegicus, one of the most economically valuable crustaceans in European waters. It is dissected in university marine biology departments. It is cataloged in fisheries surveys. It is handled, studied, photographed, and eaten in quantities that make it one of the most intimately examined invertebrates on the continent. And for the entirety of human fishing history, every net, every lab bench, every fish market from Bergnen to Barcelona, nobody noticed that an entire branch of animal life was living on its lips. In 1995, Peter Funch and Reinhard Moebjerg Christensen at the University of Copenhagen were conducting what should have been a routine microscopy session on preserved Norway lobster specimens collected from the Kattegat, the narrow strait separating Denmark from Sweden. Under magnification, they focused on the mouthparts, the labrum, and the maxillopedes, the structures the lobster uses to process food. What they found attached to the bristly lip surface defied identification. Tiny organisms, goblet-shaped, roughly half a millimeter tall. Anchored by adhesive discs, each one possessed a ciliated mouth ring, a buccal funnel, that generated microcurrents to sweep food particles from the lobster's own feeding activity into itself. They were filter feeding off the lobster's dinner scraps. Funch ran the specimens against every known classification: Cnidaria, Rotiferi, Bryozoa, Entoprocta, Platyhelminthes. Nothing matched. The body plan didn't fit any phylum in the animal kingdom. The organism was genuinely new, not a new species, not a new genus, not a new family. A new phylum. They named it Cycliophora. Only the third new animal phylum described in the entire 20th century. The species name, Pandora, was a warning. Funch and Christensen knew this discovery would open a box of phylogenetic problems that might never be fully closed. The reproductive cycle is where the horror begins. Symbion Pandora can reproduce asexually by budding an internal structure called a Pandora larva. This larva develops inside the parent body, growing until it fills the internal cavity and then bursts free. It swims briefly, hours, not days, and settles nearby on the same lobster to generate a new feeding individual, but the organism also reproduces sexually, and that pathway is worse. Dwarf males, roughly 0.08 millimeters, barely visible even under magnification, develop inside modified feeding individuals. The male embryo grows by consuming the host body from within. It cannibalizes the very individual that generated it, hollowing it out, before breaking free as a functional adult male. Its sole purpose is to locate and fertilize a female. The fertilized female then produces a chordoid larva, a sexually produced larval stage possessing a structure that resembles a notochord. When Funch first observed this, it momentarily raised the bewildering possibility that the organism might be related to vertebrates. It isn't. But the fact that it even looked plausible illustrates how alien this body plan is. Simon Conway Morris at Cambridge called the discovery one of the most remarkable of the century. Yet specimens of Symbion had been collected as far back as the 1960s by researchers examining Norway lobster mouthparts. They were misidentified as colonial protists and filed away. Funch spent over a year in continuous live observation under microscopes, documenting the complete life cycle before he and Christensen felt confident enough to publish in Nature on December 14, 1995. One detail elevates this from remarkable to existentially unsettling. When a Norway lobster molts, sheds its entire exoskeleton to grow, every Symbion colony on its body faces instant annihilation. The old cuticle peels away, taking the attached population with it. The organisms must detect the molt coming and produce Pandora larvae in advance, larvae that can swim to the fresh, naked cuticle and recolonize it before the old shell is discarded. If they fail, they are gone. Every molt is an extinction event at the colony level. Densities of up to 100 individuals per lobster have been documented on heavily colonized specimens. As of 2023, only 3 species have been described in the entire phylum. Symbion Pandora on the Norway lobster, Symbion americanus on the American lobster, Homarus americanus described in 2006, and Symbion pentabrachiatus from French waters described in 2019. 3 species. in a phylum that molecular clock estimates suggest diverged from its nearest relatives over 500 million years ago, before fish existed, before insects existed, before anything with a backbone crawled onto land. A half-billion-year-old lineage of animals was hiding on the mouthparts of something we serve with lemon wedges, and that raises a question worth sitting with. If an entire phylum can live undetected on the most commercially handled crustacean in Europe, what else is hiding, not in the deep sea, not in some unreachable trench, but on animals we already have in our hands? Number 2. Enteroxenos öStergreni, the snail that devolved into a parasite. Picture a sea snail, a coiled shell, 2 eyes on sensory stalks, a radula, the ribbon of microscopic teeth that scrapes algae and detritus off surfaces, tentacles probing the substrate, a brain, however small, a heart pumping blue blood, a functional digestive tract processing food from mouth to anus. This is what a gastropod mollusk looks like. Hold that image because what comes next erased every piece of it. In 1920, Swedish zoologist Sixten Bock was dissecting specimens of the sea cucumber Stichopus tremulus, collected from the cold, deep waters off the Norwegian coast. When he opened the body cavity, he found something that had no business being inside an echinoderm. A long, pale, convoluted tube of tissue coiling through the host's abdomen. 30 to 50 centimeters of biological material, in some cases longer than the sea cucumber itself. It had no shell, no eyes, no tentacles, no radula, no brain, no heart, no digestive tract, no nervous system, no circulatory system. What remained was a body wall, a thin outer layer surrounding a cavity packed entirely, exclusively, with eggs. Hundreds of thousands of them. It was a snail. A gastropod mollusk that had shed every defining feature of its class except the one that ensured its genes would persist: reproduction. Enteroxenos öStergreni belongs to the family Entoconchidae within the gastropod order Eulimeda, a lineage of snails that progressively abandoned free-living existence to become endoparasites of echinoderms. But Enteroxenos sits at the absolute extreme of that spectrum. It is, by any biological measure, the most drastically degenerated animal in the molluscan phylum and arguably in the entire animal kingdom. The larval stage is the proof of ancestry and the source of the horror. Free-swimming veliger larvae hatch from those hundreds of thousands of eggs with a small, functional shell and working eyes. For a brief developmental window, days, not weeks, they are genuine snails. They swim. They see. They have the equipment of their ancestors. Then, if a larva encounters a sea cucumber, it bores through the host's body wall using enzymatic secretions, enters the coulomb, and begins a transformation that takes 6 to 12 months to complete. Organ by organ, system by system, the larva disassembles itself. The shell dissolves. The eyes degrade. The nervous system unravels. The digestive tract closes and reabsorbs. What emerges at the end of this process is the organless tube that Bock found in 1920. Nutrient acquisition shifts entirely to diffusion. Enteroxenos absorbs nutrients directly through its body wall from the host's coulombic fluid. No mouth, no gut, no absorption structures. Just a permeable surface soaking up whatever the sea cucumber's body provides. Dwarf males, less than 1 millimeter, live inside the female's body cavity as permanent internal parasites, independently paralleling the same pattern seen in Bonellia viridis. Parasites within a parasite within a sea cucumber. Molecular phylogenetics confirmed in 2014 by Takano and Kano that Enteroxenos nests firmly within Eulimeda. Its closest living relatives are eulimid snails, ectoparasites that still possess shells, and feed on echinoderms by piercing them with a proboscis. The family forms a visible gradient of parasitic degeneration. The eulimids retain their shells and most organs but have lost the radula. The genus Entocalax, described by Voigt in 1888, shows an intermediate stage. It has a vestigial gut and a reduced nervous system. And then there is Enteroxenos. Total loss. The gradient is a roadmap of how a free-living snail becomes an organless reproductive tube, step by step, across evolutionary time. Ludwig von Graf suggested in 1884 that the strange organisms found inside sea cucumbers might be degenerate mollusks. The idea was dismissed as absurd by his contemporaries. It took Jens Lutzen's electron microscopy work in 1968, identifying residual muscle fibers in the body wall with the characteristic arrangement of molluscan musculature to vindicate him, 84 years after the hypothesis was first proposed. No immune response from the host has ever been documented. Enteroxenos appears to evade the sea cucumber's defense systems entirely, possibly through molecular mimicry of host surface proteins. The host shows reduced reproductive output, but no external signs of infection. From the outside, the sea cucumber looks normal. Inside, it is carrying a coiled tube of eggs the length of its own body. A single female Enteroxenos produces over 100,000 eggs. The reproductive output is enormous because the probability of any given larva encountering a suitable host is vanishingly small. The family Enteroconchidae is distributed globally, wherever their echinoderm hosts occur, from the North Atlantic to the Indo-Pacific, and Enteroxenos is not the only lineage that ran this program. An entire phylum went the same way. Number 3, Orthonectida, the Devolved phylum. Enteroxenos is one species, one snail that devolved, but there exists an entire branch of the animal tree of life where the same process occurred on a phylum-wide scale, from organisms far more complex than a snail, down to something barely recognizable as an animal. Orthonectida was first described by Elias Mechnikov in 1877 from specimens he discovered inside a dissected brittle star, a relative of starfish. The adults he extracted were microscopic, 0.05 to 0.3 millimeters. Under the microscope, their anatomy was shockingly simple. A single layer of ciliated epithelial cells arranged around a central core of reproductive cells. No nervous system in any traditional sense. No digestive system. No excretory organs. No circulatory system. Males consist of roughly 1,000 cells total. Females roughly 2,500. These are among the simplest multicellular animals on Earth by raw cell count. For over 130 years, the standard interpretation was that orthonectids were primitive. Simple organisms that diverged early in animal evolution and never acquired the complexity that other lineages developed. The textbook story was clean. They represented a window into what early multicellular animals might have looked like before nervous systems, guts, and brains evolved. This interpretation was wrong. In 2018, Mikhailov and colleagues at the Belozersky Institute of Physico-Chemical Biology in Moscow sequenced the genome of Intoscialinae, an orthonectid species that parasitizes the Nemertean worm Lineus ruber, and published their results in Current Biology. The genome was small but revelatory. It retained unmistakable molecular signatures of complex ancestral gene families, developmental signaling pathways, homeobox genes in modified but recognizable forms, the remnants of a body plan that once included muscles, a through gut, and a centralized nervous system. The approximately 70% of ancestral gene families that had been lost were not randomly distributed across the genome. The deletions targeted structural genes, neural patterning genes, and digestive enzyme genes, precisely the categories a parasite would shed when the host provides food, shelter, and everything else. Orthonectids did not start simple, they started complex. Molecular phylogenetics places them within Spiralia, the superphylum containing annelid worms, mollusks, and flatworms. Their ancestor may have resembled a marine worm with segmented muscles, a functional gut, and a brain containing thousands or tens of thousands of neurons. What exists now has 12 neurons in the female and 8 in the male. Slusarev and Staranov mapped the complete neural architecture in 2016 using confocal laser scanning microscopy. 12. 8. That is the full extent of the nervous system in an animal whose ancestors once had brains. The life cycle inside the host is what earns the word horror. Inside the body of a Nemertean worm, a brittle star, a bivalve mollusk, or a polychaete, the host range is unusually broad. The orthonectid exists as a plasmodium, a multinucleated mass, not a discrete organism but a spreading network of parasitic tissue that infiltrates the host's gonadal tissue and replaces it. The host is castrated. Its reproductive capacity is hijacked and converted into parasite biomass. The plasmodium can persist for months or years, silently expanding until it matures. When it does, it fragments. Hundreds of tiny sexual stage adults, the 12 neuron females and 8 neuron males, erupt from the host body simultaneously. The host frequently dies. The free-living sexual adults have hours. hours to find each other in open water mate, and produce larvae that must locate and penetrate a new host before their yolk reserves are exhausted. Edward Kozloff documented infection prevalences of up to 40% in Nemertine populations in the San Juan Islands of Washington State in 1969. 40% of the worms in those populations were carrying plasmodia. Castrated. Slowly being converted from the inside into vehicles for orthonectid reproduction. The phylum contains roughly 25 described species across 7 genera. But taxonomists suspect this number dramatically undercounts true diversity, because finding orthonectids requires dissecting hosts and examining their gonads under magnification. You cannot detect them from outside. George Slusarev at St. Petersburg State University is one of perhaps 5 researchers worldwide actively studying the phylum. An entire branch of animal life with a 500 million year evolutionary history investigated by a handful of people. Number 4. Osborn's pipefish and the selective embryo cannibalism. The image is iconic, a male seahorse, belly distended, releasing a cloud of miniature offspring into the water, paternal devotion, nature at its most heartwarming. The Cyngnathidae, the family containing seahorses, pipefish, and sea dragons, are famous for male pregnancy. Females deposit eggs into the male's ventral brood pouch. The male provides oxygen, osmoregulation, and nutrients through a vascularized inner lining containing prolactin receptors similar to those found in mammalian mammary tissue. Convergent evolution produced something functionally analogous to a mammalian uterus built into the belly of a fish. In 2010, Kimberly Paxholt and Adam Jones at Texas A&M University published a paper in Nature that took this heartwarming image and set it on fire. They studied the Gulf pipefish, Cygnathus scavelli, in the seagrass beds and estuaries of the Gulf of Mexico and the Atlantic coast of the United States. The experimental design was precise. Males were paired with females of varying body sizes. In pipefish, body size is the primary sexual attractiveness signal. Larger females are preferred. Clutches were implanted into male brood pouches and tracked using microsatellite DNA analysis to determine the maternity and paternity of every surviving and every reabsorbed embryo. The results were unambiguous. Males who mated with small, less attractive females reabsorbed significantly more embryos than males who mated with large, attractive females. The effect held even when clutch sizes were identical between the two groups. This was not passive pregnancy failure. It was not a resource limitation causing random embryo loss. The males were actively, selectively redirecting nutrients from specific embryos, those sired by less attractive mothers, back into their own body tissue. Deliberate filial cannibalism. Cryptic post-copulatory mate choice. The male accepts the eggs he appears to commit. And then he quietly kills the offspring he judges to be low value. Males in the Cyngnathidae can carry eggs from multiple females simultaneously within the same brood pouch. Paxholt and Jones found that males carrying mixed clutches from both large and small females selectively aborted the small female's embryos while nurturing the large female's embryos. Same pouch, same father, different fates determined by the mother's attractiveness. The resource economics make the strategy rational. A pregnant Gulf pipefish male loses 10 to 15% of his own body mass over the 10 to 14 day gestation period at typical Gulf water temperatures of 25 to 30 degrees Celsius. That is an enormous physiological investment. Males that selectively aborted less valuable embryos recovered their body condition faster and were able to enter a new breeding cycle sooner. The fitness payoff is measurable and real. Additional DNA analysis revealed that absorbed embryos sometimes had lower heterozygosity, suggesting that males may also be evaluating genetic compatibility, not merely the physical size of the mother. A follow-up study by Sage Bakin and colleagues in 2017 confirmed the phenomenon in the broad-nosed pipefish Cygnathus typhal from Scandinavian waters. Since the original publication, the behavior has been investigated in at least 7 species across the Cyngnathidae. Evidence of selective embryo abortion has been found in most of them. This is not a quirk of one Gulf of Mexico pipefish. It appears to be a widespread reproductive strategy across the family. In pipefish, sexual selection operates primarily on females. Females compete for access to male brood pouch space. It is a mirror image of typical mammalian dynamics where males compete and females choose. Here, males choose, and they choose after the fact, after the eggs are deposited, after commitment has been signaled. The decision is made in silence inside the body of a pregnant father. Number 5. Henneguya Salminicola, the animal that stopped breathing. Every animal on Earth uses oxygen. This has been treated as a biological law since the acceptance of Lynn Margulis's endosymbiotic theory in the 1970s. Mitochondria, the organelles inherited by every animal cell, process oxygen through aerobic respiration to generate ATP, the universal energy currency of life. From a blue whale to a nematode to a sponge, every animal possesses a mitochondrial genome that encodes the molecular machinery for this process. It is as close to a non-negotiable rule as biology has. In 2020, a team led by Dorothée Huchon at Tel Aviv University published a paper in the Proceedings of the National Academy of Sciences that broke the rule. Their subject was Henneguya Salminicola, a myxozoan parasite found embedded in the muscle tissue of Chinook salmon and coho salmon in Pacific Northwest river systems. Using deep sequencing, fluorescence microscopy with mitotracker staining, and comprehensive genomic analysis, the team demonstrated that Henneguya Salminicola has completely lost its mitochondrial genome. Not reduced it, not modified it, lost it. It is the first and, as of 2025, the only known multicellular animal that cannot perform aerobic respiration. It does not breathe oxygen. It is the animal that stopped being an animal by the one metabolic criterion we thought was universal. The organism retains organelles derived from mitochondria, but these have been repurposed. They no longer perform oxidative phosphorylation. The working hypothesis, published by Yahalomi and colleagues in the same research group, is that the parasite imports ATP directly from the host cell's cytoplasm using specialized transporter proteins. It steals its host's energy rather than generating its own. An alternative is that it derives energy through anaerobic metabolic pathways not yet fully characterized. Fishermen in the Pacific Northwest know Henneguya Salminicola by a different name. They call it tapioca disease. Infected salmon flesh is studded with milky white cysts, pseudocysts, that make the meat visually unacceptable for commercial sale. The fish is not harmful to eat, but the appearance renders it worthless. In some British Columbia river systems, infection prevalence reaches 90%. Annual losses to Pacific Northwest salmon fisheries run into millions of dollars. The parasite is distributed from Alaska to California. Myxozoa, the broader group to which Henneguya belongs, are themselves one of the great biological plot twists of the last century. For most of the 1900s, they were classified as protists, single-celled organisms. It was not until molecular phylogenetics in the early 2000s confirmed that Myxozoa are actually cnidarians, relatives of jellyfish and corals. The proof is in the spores. Henneguya spores are 10 to 20 micrometers long and possess polar capsules with coiled tubules. These structures are homologous to the stinging nematocysts of jellyfish. An entire clade of jellyfish relatives had compressed themselves into microscopic parasites, lost nearly every feature of their free-living ancestors, and infiltrated the muscle tissue of fish. The complete myxozoan life cycle, which involves alternating between a fish host and an annelid worm host, was only fully elucidated in the 1980s. Huchon's team compared Henneguya Salminicola with a closely related species, Henneguya shokae, which retains a functional mitochondrial genome. The loss of aerobic respiration is specific to Salminicola's lineage, a recent evolutionary event, not an ancient one. The genome has been reduced to approximately 16.7 megabases, a dramatic compression from the 300-plus megabase genomes of free-living conidarians. Evolution took a jellyfish, compressed it into a microscopic spore, stripped away its tentacles, its bell, its nervous system, and then removed the most fundamental metabolic requirement of animal life. Some biologists have argued the finding necessitates updating the formal definition of what an animal is. Number 6, the Loricifera of l'Atlante Basin. 3,500 meters below the surface of the Mediterranean Sea, approximately 200 kilometers west of the island of Crete, the seafloor drops into a depression called the l'Atlante Basin. It is not an ordinary patch of deep seafloor, it is a lake, a literal lake, on the bottom of the ocean. Brine with salt concentrations 5 times higher than normal seawater pools in the depression, so dense that it does not mix with the water above. It forms a visible body of water with a discernible shoreline, an underwater lake complete with a surface you could theoretically stand on. The brine is anoxic, completely permanently devoid of molecular oxygen. It has been this way for at least 50,000 years. By every conventional understanding of animal biology, nothing multicellular should be alive in it. In 2010, Roberto D'Annavaro and his team at the Polytechnic University of Marche in Ancona, Italy, published findings in BMC Biology that redrew the boundaries of what animal life can tolerate. Across sampling campaigns spanning 1998 to 2008, they collected over 700 specimens from the basin floor. Among them were 3 new species of Loricifera, microscopic animals belonging to 3 separate genera, Spinoloricus, Rugiloricus, and Plissiloricus. They were not dead specimens that had drifted down from oxygenated waters. They were alive. Rose Bengal vital staining confirmed active metabolism. Cell tracker green, CMF. DA fluorescent markers confirmed intact cellular processes. Transmission electron microscopy revealed intact ultrastructure, and some specimens contained eggs in active reproductive states. These animals were completing their entire life cycles, feeding, growing, reproducing, in permanent anoxia. Loricifera is a phylum most people have never heard of. Discovered in 1983 by Reinhard Christensen, the same researcher who later co-discovered Symbion Pandora's phylum Cycliophora, Loriciferans are microscopic marine animals, 100 to 400 micrometers long, with an ornate external shell called a lorici and a retractable head structure called an introvert, armed with spines and scalids. They live in marine sediments worldwide and require specialized meiofauna extraction techniques to collect. They are rarely studied because they are tiny, difficult to find, and uncharismatic by any popular standard. The Lattilante loriciferans were different from all previously known species in one critical respect. Electron microscopy revealed that their cells lacked functional mitochondria. In their place were hydrogenosomes, double-membraned organelles that produce molecular hydrogen and ATP through anaerobic pathways. Prior to this discovery, hydrogenosomes were known only in single-celled organisms, particularly anaerobic protists like Trichomonas. No multicellular animal had ever been documented using them. These loriciferans had replaced the fundamental energy processing machinery of animal cells with something that was supposed to exist only in protists. The finding was initially challenged. Alexander Chesunov of Moscow State University questioned whether the stained specimens were truly alive or merely recently deceased organisms preserved by the anoxic conditions. D'Annavaro's team responded in 2016 with additional evidence, including specimens captured in active reproduction. The debate was settled. The implications extend well beyond taxonomy. If multicellular animal life can persist in anoxic hypersaline brine for 50,000 years or more, using metabolic machinery borrowed from protists, the conditions for complex life in the universe expand dramatically. The subsurface ocean of Jupiter's moon Europa and the hydrothermal vents of Saturn's moon Enceladus, both anoxic, both saline, become more plausible habitats for animal-grade life. The Loricifera of Lattilante Basin are not just a biological curiosity, they are a data point in the search for life beyond Earth. Number 7. Bonellia viridis, the green spoonworm. In the rocky crevices and understones across the Mediterranean and the eastern Atlantic, from shallow subtidal zones to depths of roughly 100 meters, there lives a vivid green worm. The female Bonellia viridis is 6 to 8 centimeters long. Her proboscis, a long forked feeding appendage, can extend up to a full meter from her crevice, sweeping across the seafloor to collect detritus. She is bright green because of a pigment called bonaline, a chlorine derivative that doubles as a potent cytotoxin and photosensitizer. Laboratory assays have demonstrated bonaline's antimicrobial, antifungal, and anti-tumor properties. She is, by the standards of marine worms, an unremarkable detritivore with an unusual color. The males are 1 to 3 millimeters long. They live inside the female's nephridia, excretory organs that double as reproductive ducts. Their digestive systems are gone. Their nervous systems are simplified. Their sole biological function is to produce sperm. Up to 20 or more males can inhabit a single female simultaneously, packed into her reproductive tract as permanent internal passengers. They will never leave. They will never feed. They will never do anything except provide sperm until they die. The mechanism that creates these males was first described by Fritz Balzer at the University of Bern in 1914 and confirmed through a series of increasingly elegant experiments over the following 2 decades. Free-swimming trochophore larvae hatch from the female's eggs and enter the plankton. They are sexually undifferentiated. They have no assigned sex. For 2 to 6 weeks, they float, feed, and grow, carrying the potential to become either sex. Their fate is determined by one event: a larva that settles on the seafloor and contacts rock, sediment, shell, or any substrate that is not a female Bonellia develops into a female. Over 1 to 2 years, it grows, matures, turns green with bonaline, and takes up residence in a crevice. A larva that contacts a female's proboscis is exposed to bonellin. Within 24 to 48 hours, masculinization begins. The larva shrinks. Its digestive system shuts down. It develops primarily as testes. It migrates toward the female's body opening, is absorbed into her reproductive tract, and is sealed inside. The transformation is irreversible. Laboratory experiments later proved that purified bonellin applied to larvae in the absence of any female still triggers male development. The molecule alone is the switch. The sex ratio in natural populations is heavily female biased. Males are produced on demand, only when a larva physically contacts a female. This is local mate competition avoidance taken to its logical extreme. Do not waste energy producing males unless a female is already present to house them. The evolutionary efficiency is ruthless. One touch of a toxin on a stranger's skin, and your body plan, your lifespan, your size, your autonomy, and your biological identity are permanently rewritten. Number 8. Dimorphalus gyrociliatus, the disposable male. Females are normal, 1 to 2.5 millimeters long. Functional digestive system with a mouth, pharynx, and gut. A nervous system containing 300 to 500 neurons. Reproductive organs capable of multiple breeding cycles. A typical small polycatianellid worm found in intertidal algal mats and brackish sediments around the world. Males are 0.3 to 0.5 millimeters. They have no functional mouth, no pharynx, no digestive tract of any kind. They cannot eat. They survive on yolk reserves deposited in the egg before they hatched. They were born with a fixed fuel supply and no means of refueling. Their lifespan is 1 to 5 days after emergence. During that window, 1 to 5 days, the male's sole activity is to locate a female, attach to her body, transfer sperm through a copulatory stylet, and die. Its body is dominated by a single oversized testis that fills most of the body cavity. Its nervous system contains fewer than 100 neurons. Its sperm are a flagellate and amoeboid. They do not swim. They crawl, dragging themselves forward through the low-flow microenvironment between sediment grains. Males mature sexually within hours of hatching. They emerge from the egg. Functionally adult reproductive system fully operational. Countdown already started. Sex determination is visible before birth. Female eggs are large, roughly 90 micrometers in diameter. Male eggs are small, roughly 50 micrometers. Both types are visible within the mother's body. A typical clutch contains 2 to 5 large female eggs and 5 to 15 small male eggs. The males outnumber the females because they are expendable. More males means higher probability that at least one finds a female in the chaotic intertidal environment before its yolk runs out. The species was first named by Otto Schmidt in 1857. Oscar and Cécile Vogt documented the extreme dimorphism in the early 1900s. More recently, Martin Duran and colleagues published genomic work in 2021 using Dimorphalus gyrociliatus as a model organism for studying nervous system evolution. The genome is approximately 73 megabases, one of the smallest known animal genomes, comparable to the nematode C. elegans. The compact genome reflects the overall miniaturization of the organism. Laboratory cultures are maintained at the University of Bergen and the Max Planck Institute for Biology in Tübingen. The genus Dimorphalus contains at least 5 species, all showing some degree of sexual dimorphism. But Gyrociliatus is the most extreme. The female reproduces many times across her lifespan. The male reproduces once. These are opposite survival strategies, Iteroparis versus Semmelparis, encoded from the egg onward in the same species. The disposable male is not a defect, it is a design specification. Number 9. Xenoturbella. the unclassifiable worm. In 1949, Swedish zoologist Einar Westblad examined specimens dredged from the Gullmarsfjord on Sweden's west coast and found himself looking at something he could not place in the animal kingdom. 1 to 4 centimeters long, pinkish-brown, soft-bodied, featureless, a sock-shaped blob with a subtle longitudinal furrow and absolutely nothing else that could serve as a diagnostic feature. No brain, no through gut, just a blind-ended gastric pouch, no anus, no eyes, no discrete reproductive organs, no coulomb, no excretory system, no respiratory structures. The organism moved by ciliary gliding, its entire epidermis coated in cilia that propelled it slowly across the muddy substrate. Westblad placed it in its own family, Xenoturbellidae. He admitted openly that he could not assign it to any known phylum with confidence. Over the following decades, various researchers tried. Some classified it as a turbellarian flatworm. Others proposed it was a primitive deuterostome distantly related to vertebrates. Nobody was sure. In 1997, a molecular study by Noreen and John Delius appeared to solve the mystery. DNA analysis placed Xenoturbella squarely within mollusca. It was apparently a radically degenerate clam relative. The finding was published, cited, and absorbed into the literature. It was also completely wrong. In 2003, Borlaug and colleagues repeated the molecular analysis, this time carefully extracting DNA from deep body tissue rather than from regions near the gut. The mollusk sequences vanished. The earlier study had been contaminated by DNA from the nucula clams that Xenoturbella eats. Clam DNA had permeated the animal's tissues, and the first molecular analysis had sequenced the organism's lunch instead of the organism. For 6 years, a deep-sea worm had been misclassified because it ate clams, and scientists sequenced the clam. Modern phylogenomic analyses by Cannon and colleagues published in Nature in 2016 placed Xenoturbella within a new phylum, Senecoelomorpha, alongside aquelomorph flatworms. But its exact phylogenetic position remains one of the most actively debated questions in animal systematics. Some analyses place Senecoelomorpha at the base of Bilateria, making Xenoturbella the most primitive bilaterally symmetric animal on Earth, a living fossil of the body plan that eventually gave rise to everything from insects to humans. Others place it within Deuterostomia, closer to vertebrates, which would mean it once had a brain and a gut and a complex body plan and lost all of it. Secondarily simplified, devolved. In 2016, 4 new species were described from deep water off Monterey, California. Xenoturbella churro, named for the Spanish pastry it resembles in cross-section. X. monstrosa, at approximately 20 centimeters, the largest known species. X. profunda. And X. hollandorum. All were found at depths between 1,700 and 3,700 meters, near chemosynthetic bivalves at cold seeps. The Gullmarsfjord population is now monitored by researchers at the University of Gothenburg using specialized trawls at 30 to 100 meters depth. Xenoturbella possesses a unique statocist, a gravity-sensing organ unlike any found in other animal groups. Its nervous system is a diffuse net embedded in the epidermis with no centralization, no brain, no ganglia, no nerve cords. Its reproduction is almost entirely unknown. Fertilized eggs have been found in the vicinity of adults. That is the sum total of current knowledge about how it mates or develops. An animal that humiliated molecular phylogenetics, defied classification for 7 decades, and may or may not have once possessed a brain. Number 10. Gyrodactylus salaris. The salmon skin plague. Norway's Atlantic salmon rivers are ecological and cultural treasures. Among the most pristine freshwater systems in Europe, they sustain wild salmon populations that have been fished, celebrated, and managed for centuries. Beginning in the late 1970s, salmon populations in river after river began to collapse, not small declines. 85 to 98% losses. Entire cohorts of juvenile fish, par, disappeared. Year classes failed to recruit. Rivers that had supported abundant salmon runs for thousands of years went quiet in the span of a single decade. The culprit was a flatworm smaller than a grain of sand. Gyrodactylus salaris is a monogenean ectoparasite. 0.3 to 0.5 millimeters long, first formally described by Tor öStergreni and colleagues at the University of Oslo in 1986. It attaches to the skin, fins, and gills of Atlantic salmon using a posterior attachment organ called the opisthopter, armed with 16 marginal hooks and 2 central anchors that pierce the host's epidermis. It feeds on mucus and epidermal cells. One parasite does negligible damage. Thousands of them strip the protective mucus layer, expose underlying tissue, create open wounds, and invite secondary bacterial and fungal infections that overwhelm the fish's immune system. The parasite originated in Baltic salmon populations, where it is relatively benign. Millions of years of coevolution between Baltic Salmo salar and Gyrodactylus salaris produce partial host immunity. Baltic salmon mount an effective inflammatory response that limits parasite numbers. The introduction to Norway was entirely human-caused. Infected salmon smolts from Swedish and Finnish hatcheries were stocked into Norwegian rivers during the 1970s and 80s as part of fishery supplementation programs. Norwegian Atlantic salmon had no evolutionary history with the parasite. No co-evolved immunity, no defense. The reproductive biology is what transforms Gyrodactylus salaris from a nuisance parasite into a biological weapon. It gives live birth. The mother produces a single, fully developed daughter through viviparity. That daughter, at the moment of birth, already contains a developing granddaughter inside her. 3 generations, mother, daughter, and granddaughter, are nested simultaneously inside a single organism. Combined with the capacity for parthenogenesis, reproduction without mating, a single parasite arriving on a single host can generate an exponentially growing population entirely on its own. No mate required, no sexual reproduction required, just division, birth, and immediate preloading of the next generation. The generation time is 3 to 5 days at the parasite's optimal temperature range of 6 to 12 degrees Celsius. One parasite becomes thousands within weeks. Juvenile salmon, par, that spend 1 to 4 years in freshwater before migrating to sea are the primary victims. Adults that migrate to the ocean shed the infection because Gyrodactylus salaris cannot survive in seawater above 7.5 parts per thousand salinity. But par are trapped. They live in freshwater year-round. They are continuously exposed. In heavily infected rivers, par mortality reached 100%. No juveniles survived to become adults. The population could not replace itself. Norway's response was extraordinary by any standard. The government committed over 1 billion Norwegian kroner, approximately $100 million to eradication programs using rotenone, a plant-derived toxin extracted from the roots of tropical legumes. Rotenone kills all gill-breathing organisms in treated water. The eradication protocol requires poisoning an entire river system, killing every fish, every invertebrate, every gill-breathing organism to eliminate the parasite along with its hosts. The treated rivers are then restocked with clean salmon from uninfected hatcheries. As of 2023, approximately 17 rivers have been declared successfully treated, with salmon populations recovering. But by the time the full scope of the crisis was understood, the parasite had been detected in 46 Norwegian rivers. Alternative treatments, including aluminum sulfate, which damages the parasite's attachment hooks without killing all aquatic life, have been trialed with partial success. Genetic studies by Hansen and colleagues in 2007 identified at least 7 distinct mitochondrial lineages of Gyrodactylus salaris, some pathogenic to Atlantic salmon, and others apparently harmless. The International Council for the Exploration of the Sea lists the species as one of the most significant threats to wild Atlantic salmon populations in the world. An entire nation poisoned its own rivers systematically, deliberately, to fight a flatworm that arrived because humans move fish from one river system to another without checking what was writing on their skin. These 10 organisms share something more fundamental than their obscurity. They are evidence of what happens when you stop projecting human assumptions onto the natural world and look at what evolution actually produces. Multiple entries on this list are not stories of evolution building complexity. They are stories of evolution tearing it down. Enteroxenos shed every organ that made it a snail. Orthonectida descended from complex worm-like ancestors and stripped themselves to parasitic cell clusters containing fewer than a dozen neurons. Henneguya Salminicola abandoned the single most fundamental metabolic requirement of animal life. These are not failures. They are not degenerations in the pejorative sense. They are optimization. When a host provides food, shelter, transport, and energy, every organ that duplicates those services becomes a metabolic liability. Evolution removes liabilities. It does not care whether what remains looks like an animal by our standards. It cares whether what remains reproduces. Dollo's law, formulated in 1893 by Belgian paleontologist Louis Dollo, states that complex structures, once lost in evolution, are almost never regained. The genomes of these organisms confirm the law. Pseudogenes, broken, non-functional remnants of once active genes, litter their DNA like ruins of demolished buildings. Orthonectida's genome retains the faint signatures of a complex ancestor, the way a vacant lot retains the foundation of a house. The genes that built the brain are still there. They just don't work anymore. The reproductive strategies are equally extreme, and they share a common driver. The ocean is a three-dimensional fluid environment where finding a mate is far harder than on land. Bonellia viridis solved this by not producing males unless a female is already present. Dimorpholus gyrociliatus solved it by mass-producing disposable males so cheap that their loss is immaterial. Gyrodactylus salaris solved it by eliminating the need for mates entirely. A single individual carrying 3 preloaded generations can colonize a new host alone. And the pipefish added a chilling refinement. Even when paternal care exists, it is conditional. Not all offspring are valued equally. The father decides, in silence, which embryos live and which are absorbed back into his body. These organisms are absent from science documentaries for reasons that have nothing to do with their importance. They are microscopic. They are hidden inside hosts. They are invisible to cameras. The horror they embody is conceptual. It requires understanding mitochondria, phylogenetics, and developmental biology to appreciate. Documentaries prioritize charismatic megafauna because large moving animals in good lighting drive viewership. A half-billion-year-old phylum discovered on lobster lips does not. Funding follows attention, attention follows cameras. The result is that organisms holding keys to astrobiology, pharmacology, and the fundamental definition of animal life receive less public exposure than a single pod of orcas. The deep ocean remains the least explored biome on the planet. An estimated 80% of marine species have not been formally described. Environmental DNA sampling routinely detects genetic signatures of organisms that have never been collected, named, or studied. For most of the organisms in this video, basic life cycle data, how they mate, how they develop, how long they live, is incomplete or entirely unknown. Entire phyla are investigated by fewer than 5 researchers worldwide, and the habitats these organisms occupy, cold seeps, anoxic basins, hydrothermal vent fields, hadal trenches, face increasing pressure from deep-sea mining proposals that may destroy ecosystems before anyone catalogs what lives there. The ocean does not build what we expect. It does not respect our definitions or our comfort. It produces snails that devolve into bags of eggs, phyla that reverse engineer themselves into cell clusters, animals that abandon breathing, fathers that cannibalize their own offspring based on their mother's attractiveness, worms that have no brain and fooled molecular biology for 6 years by eating clams. These are not the strangest things in the ocean. These are the strangest things we have found so far. For every organism on this list, there are likely hundreds that have never been sampled, never been sequenced, never been named. The ledger is nowhere close to balanced, and every year it grows.